Thanks, Peiwa, and thank you all for coming to the talk today. It's really a pleasure to be invited here to Monell. This is the first time I've visited, and it's such a it's such a unique place to talk. Obviously, normally. When I give talks at different places, I have to explain, so much of the background and establish, why I'm interested in taste and smell. Here, I mean, this is a given. But hopefully you'll appreciate some of the things that we're going to do today. It's been a pleasure talking to many of you this morning, and I hope to continue our conversations throughout the day. So today I'm going to be discussing a little bit about our work investigating the dynamics of innervation and synapse formation in the taste bud. So obviously, usually I start with this: why study taste? And I have to kind of fight for why why we study taste. Thankfully, with this audience, I do not have to make such an argument. So I'm going to switch this to a different question: why do I study taste? I study taste. I'm a basic biologist at heart. I find the sense of taste to be very interesting, extremely gratifying, and full of so many questions that I'm eager to answer. And so the beauty of the taste system for me is that it is simple. We have 5 basic taste qualities that we can detect on our tongue. And as I was saying to Peiwa this morning, as soon as things get into the brain, it gets too confusing, and I kind of give up. But I feel that I can make significant progress in understanding more basic things at the periphery, understanding how these chemicals within our food are detected, how those signals are transformed from chemical to electrical signals through synapses in the periphery and leading to the brain. And hopefully by understanding this process in a more simple, although albeit delicate and wonderfully balanced way, we can understand more about basic functions of taste synapse development and plasticity in a real tractable model. Because, we can use mice to understand the taste system, and it's very similar to that of humans. Human babies, this is my PhD student Shannon's baby when she was just starting to eat. She was provided some mashed beans, and she really loves mashed beans. They have a nice umami, maybe slightly sweet, maybe slightly salty flavor, and she's really into that. And similarly, if you give a mouse a sweet tastant on a licking port, they will readily lick and, seem to enjoy it, right? Similar to babies. On the other hand, if you give a baby something that's a little bit sour, like a lemon, you'll taste it, start giving it oral facial reflex. That is very distinctive and highly obvious what's happening there. And mice do the same thing. Although mice cannot vomit, they do this orofacial gagging reflex, the gape reflex that we can observe. And because of these, highly stereotypical, very hardwired types of reflexes to taste stimuli, aversive and attractive, I find this to be a great model system to understand some of the fundamentals of the biology of the taste field. And of course, it gives us really important information about the types of foods that we're eating, whether they're going to give us nutritional value, or maybe they might be toxic or should be avoided. And so the appropriate connectivity of the peripheral taste system is really the very first aspect of making this decision point of whether to eat and accept something or to reject it. So I know you all are very well versed in the anatomy of the taste system, but I wanted to highlight some of the beautiful artwork that was done by one of my PhD students in the lab, Kimberly Baker, when she worked together with my student Shannon Landon on a recent review paper. So in this diagram, she's illustrating the different nerves that are innervating the tongue. We have in the front of the tongue the fungiform papilla that are innervated by the chorda tympani nerve here that comes around, and forms the cell bodies which are in the geniculate ganglion. The glossopharyngeal nerve innervates the circumvallate papilla and the foliate papilla in the back. The glossopharyngeal nerve innervates the palate. And in the fujiform taste buds, like this one represented here, it's a collection of taste receptor cells and also a resident population of stem cells at the base of the bud that repopulate the taste receptors. So one of the most fascinating aspects, I think, of the taste system is that the taste receptor cells are constantly turning over. And there's this resurgence of taste receptor cells being born, developing, integrating within the circuit. And the fact that these ganglion neurons that are depicted here have to find partners to carry that taste information from the receptor cell to the brain through the ganglia. So this process is constantly turning over, reproducing all the taste receptor cells, and within a month's time, pretty much the entire population of the taste bud has been replenished. So although, the taste receptor cells have changed from month to month, we all know that our sense of taste remains fairly consistent across that time. If you taste some bitter coffee in the morning today, next month, it'll pretty much taste the same amount of bitter. So there has to be this reconnection and rewiring consistently over time. And so this dynamic question was really intriguing to me, and there's been a lot of work done by members of this audience to figure out some of the very important aspects of how this is maintained. So over years of work, I've identified that there's trophic factors that help maintain peripheral taste integrity. These factors come both from within the taste bud environment and also the ganglion neurons. And Dr. Zhang's lab has made incredible progress identifying arspondin as one of these trophic factors that is absolutely necessary to provide signals to the proliferating taste stem populations to regenerate the taste bud. Without these signals, the taste bud kind of stops populating and becomes dormant. So how do nerve fibers find and maintain synaptic contacts with the taste receptor cell? We've come on this question to look more in depth about how taste synapses are formed. We know that the gustatory nerve is providing trophic factors to maintain some of the taste bud homeostasis, but is it also important for developing sites of contact and sites of synapse between taste receptor cells and taste neurons? So this is one of the questions that we're really interested in following up on in the lab. So in a broader picture, my research questions in the lab is to investigate the connectivity, plasticity, and function of peripheral taste circuits in health and in disease. So I kind of break this down into three different areas. One is connectivity. What are the synaptic partners of specific taste receptor cell types? Do they form connections between specific cell types to preserve that kind of sweet to sweet, bitter to bitter connections that we had initially identified using the semaphore model? Another question we want to answer is plasticity. How do taste synapses change during taste turnover? Obviously, as the cell dies, the gustatory nerve is left without a partner and then might need to find a new partner when new taste receptor cells are maturing and identifying itself into the bud. And then also in the midst of this, we want to identify how age, disease, or diet can affect peripheral taste connectivity and function. So these are the very broad aspects of the research that's ongoing in my lab. And we've only delved into a little tiny bit of each of these, but I wanted to highlight some of those questions that we're looking at and how we're developing tools to get answers to those questions. So we figured we need some extra tools to explore the connectivity and plasticity of the gustatory system. And one of these tools that I've been working on is called GRASP. So this is a project that was spearheaded by Shannon Landon, whose baby was the featured actor in the beginning here. Shannon was a recently graduated student. She's now at WashU in St. Louis pursuing her postdoc. So we were really interested in using the GRASP system, which I had initially come across in when I was in the Zucker lab, and we had tested out some constructs in Drosophila to figure out how to make GRASP different colors, how to make it activity dependent. But I really initially wanted to work on the GRASP system to answer this question in the taste field, like who is connecting with who in the taste bud. So Shannon helped develop some of these tools that we're now working on. So the idea behind GRASP is that it's a GFP molecule that's been split into two pieces. So there's one part of the GFP gets placed on a presynaptic cell, and the other part of the GFP, which can be very small, it's actually just one of the 11 beta barrels within this beta, or one of the beta sheets within this beta barrel, is expressed on the postsynaptic side. So separately, these two pieces of GFP do not fluoresce, but as soon as they come into close contact, such as at a synapse, they can spontaneously refold and form a fluorescent molecule that you can see with light microscopy. So the idea behind this is to put GRASP into the taste system to probe where the taste receptor cells and taste ganglion neurons synapse, and hopefully be able to use this to monitor taste synapse formation, reconnection, and coming apart and back together again over time. So to do this, we needed to take advantage of both transgenic and viral approaches. So unfortunately, as I was discussing with Guillaume this morning, the taste receptor cells, because they turn over so quickly, they're not really amenable to viral-based tools. I mean, you need them to be very quickly induced. So we figured the best way to get GRASP, the presynaptic GRASP to be expressed in taste receptor cells is through a transgenic mouse. So we engineered several transgenic mice to do this. And then we also have a viral approach to infect the geniculate ganglion through expression through the nucleus of the solitary tract. So we have a retrograde type of virus that is able to infect those ganglion neurons and express broadly throughout the geniculate ganglion. We also have made certain viruses where we can use Cree dependence to put that postsynaptic side of the GRASP in subsets of ganglion neurons driven by a Cree driver. So here's one example of the pre-GRASP mouse that we made that it ends up expressing GRASP in all of the taste receptor cell types. So Shannon looked at the GRASP, which is expressed, and we can use immunohistochemistry versus with the anti-GFP antibody to find out where GRASP is expressed. In this mouse, this is a pan-TRC GRASP where we can see expression in both the PLC beta 2 cells here in red, the CAR4 cells here looking yellowish and co-expressing with green, and then NTPDAs staining the type 1 cells. So we can see that in all of the types 1, 2, and 3 cell types, we can get GRASP expression in all of those guys.
Do you mind questions for
Oh, yes, go ahead, please.
So it looks like plasma membrane localization.
Yes.
What is you're referring to the GFP link to?
Yeah, so in this case, it's a neuroligin. Yeah, so we have a neuroligin little mini peptide, like a signal sequence type of thing, and it's linked to there. Or not single sequence, it's linked to the transmembrane and intracellular portion of it.
Is neuroligin normally expressed in all the cells?
So when I did like a RNA sequencing profiling of taste buds, neuroligin is quite well expressed in bitter. It's not as highly expressed in the other cell types, but it seems to be working well enough that it's getting to this membrane. So it's not hindering its localization, but in the GRASP system, where this is normally done in the central nervous system, the neuroligin helps get it to the actual synaptic compartment rather than just generally on the membrane. Here we see it essentially coating the membrane. I had made a previous version of GRASP which had just a CD4 linker, so it would just go generally membrane. We don't see a huge difference between the neuroligin bound version versus the CD4 version in our hands. But I think in the CNS, it probably does a better job in getting it to the pre and the post synapse. So on the other side would be Milrexin.
Here it doesn't like it's doing that,
No.
right? And it's going to plasma membrane.
Yeah, yeah, it's coding to the plasma membrane. So it's not localizing specifically in a presynaptic specialization. So yeah. Yeah. So in this mouse, we have got a version that it's pre-GRAS is expressed in pretty much all of those taste receptor cell types. And we have seen grasp happening at contact. So this is an example of the red is labeling the gustatory neurons innervating the CV here. And then right at these tips, you can see some green labeling happening. This is less than I was hoping for, right? I was hoping for a lot more grasp here. And this is one of these mice where it's should be expressed, the pre-GRAS should be expressed on most of the cell types. So this is a little bit less than I was expecting. I don't know whether or not this is because the GRAS works only in type 3 cells, or if I'm just not getting high enough labeling and enough of those GRASP molecules to combine together to get a bright enough signal. So we're still working on some of the, the nitty gritty of improving expression on both sides of the cells and seeing whether we can localize these to both type 2 and type 3 synapses. We have, yeah,
sorry.
Go ahead.
The red is a neuroligand receptor. That's why you expect more of the.
So no, actually the red is the postsynaptic grasp, which the virally induced postsynaptic grasp in the gustatory neurons innervating these circumvallate papilla cells. So the entirety of the gustatory neuron is labeled in red. So we can see all of the fibers that we're expressing.
But that's part of the deal because the patents aren't being sold.
Yeah.
Attached to what?
Neurexin. So I forgot Neurexin, yeah. And I might have this backwards. It might be Neurexin neuroligand. I forgot one is post and one is pre, and I'm like having a flipping movement, but yeah, it's one or the other. I can get back to you with the details.
Sorry if I missed it, but
Yes, go ahead.
how do you express the part that's in the taste bud? Is that viral as well?
No, so that's a transgenic mouse. That's the problem, right? We couldn't just get it expressed virally. If I could, I would, because it would make my life much easier. Unfortunately, because of the turnover time of the taste receptor cells and just getting it to express in specific cell types, we wanted to make a transgenic mouse. So this is transgenically expressed pre-GRASP in the taste receptor cells and virally expressed post-GRASP in the gustatory neurons.
Thank you.
So in a different mouse, we were able to get a different color of GRASP. This is the cyan version of GRASP rather than this one is actually a yellow fluorescent protein version of GRASP. In this mouse, we use PK8201 to drive expression of the pre-GRASP in the sour cells. And we could then express the postsynaptic GRASP with the viral tool like we just did in the postsynaptic side. And we could see really beautiful looking puncta here. This is in using a 2-photon approach. So we can actually see these in a live mouse without killing the mouse and image them in real time. So in that version, we're using PKD201 to drive the GRASP in the sour cells, and we can see it even in a live mouse with a 2-photon microscope. In addition to sectioning and. Okay. So one last little thing that we've been working on is tissue clearing and light sheet microscopy. This is just a fantastic picture. I just wanted to share with you because I think you would appreciate. So this is another FOX2B AI9 mouse line that we've just taken the whole tongue. Actually, this is just the 2/3 front, so we haven't included the CV. We have a CV block in the back that we have also imaged. But it's just amazing to me, the level of detail that you can see. And this is only with a 5x objective lens. We've gone through and imaged the front of the tongue and the more of the interior portion with a 20x, and you can actually see individual fibers innervating these taste buds. And these are the taste buds. out here sticking out. So we've done a few of these light sheet images, and they take up terabytes of space on your computer. So it's kind of not the easiest thing to work with as far as data, but it just absolutely produces such beautiful images. And so now that we're more interested in looking at recovery after nerve cut, these are types of techniques we plan to use in the near future to see how long does it take for the nerve fibers to reinnervate. How do they reinnervate? What's the branching pattern? Is it similar or different to what like the normal status is in the mouse? Okay, so I know I've taken up so much time just because of technical difficulties, so I might not get through both of these. But these are two papers that are either in process or have been recently published in my lab, investigating the effects of chemotherapy on innervation in the taste bud and investigating the role of innervation in developing or maintaining taste synapses. And these are two projects that were headed up by my two latest PhDs in the lab, Shannon Landon and Ryan Wood, along with my other technical and master's student support. So as you all know, chemotherapy really affects the taste system, and over a third of patients undergoing chemotherapy treatment experience taste dysfunction. And actually, I hadn't realized this until I started giving talks to the general public, and people would come up to me after talks and probably have had the same experience, saying, oh, I either went through chemotherapy or my husband or my wife or whoever went through chemotherapy and they lost their sense of taste, and this is really disturbing to them. And in the literature, you can find that this distorted sense of taste is really linked to poor recovery and a reduced quality of life, and has been indicated as one of the more severe side effects of going through chemotherapy treatment. So a lot of work had been done to date of looking primarily at the effects of chemotherapy on the taste receptor cells. And it has been well known that the chemotherapy has both a cytotoxic effect on the taste receptor cells at higher dose of chemotherapy, but also it pauses the regenerative capacity of the proliferating progenitor cells. But one thing that we hadn't seen in the literature to date was how chemotherapy might affect the gustatory neurons innervating the taste buds. And so we looked at that a little bit more carefully. And so we started just imaging using doing a whole amount immunofluorescence study and looking at these fibers. And we saw that with FOXGB-EI9 lines, after chemotherapy, I think it was 100 mg per kg dose after 3 or 4 days, I can't remember, I'm sorry. There was a significant reduction in the volume of gustatory fiber innervation within the bud. We also saw a reduction in trauma staining, keratin staining within the bud, as we had expected. But actually, the amount of gustatory fiber innervation that was lost was a little bit more significant than the taste receptor cells at this point. So we wanted to take a more in-depth view to see what is actually happening in real time. And so we performed this 2-photon imaging experiment, and in control lines, we can image the same buds over days. And we see very little changes in the volume of fibers on average. There's some increases, there's some decreases. Yes, there is this dynamics that's happening, but they tend to maintain, on average, about the same amount of innervation over time. And we don't see any effects or bad effects of the imaging process on the gustatory fibers.
You know that these are gustatory fibers because
they are.
You see them ending in taste buds, or
Yes, but also because we're using the FOX2B Cree line, which has been shown to be primarily gustatory. There might be some somatosensory in there, maybe, but primarily gustatory. So if we give a mouse saline, like we were saying before, we can image them every day or every couple of days and see a fairly consistent level of innervation. But if we inject the mouse with cyclophosphamide, we can see that these buds tend to reduce their innervation, but it's a bit variable. There's some buds that seem to keep how many fibers that were there or remain the same, that blue one at the top, but the majority of buds lose innervation and go quite and have a significant reduction in innervation over time. And so this is just a more quantification of this. Compared to day 0, after day 4, there's a significant drop off in the volume of gustatory fiber innervation in these taste buds. And just to visualize this, this is the same bud image on day 0 before injection and day 4 after the cyclophosphamide injection. And these are tracking around the same Z frame. And so you can see the biggest effect is kind of at that point where the more dorsal region of the buds have reduced their innervation. At the core and the base of the bud, it remains mostly the same. And so we think that this indicates that the fibers are either retracting down to the core or losing, or they might be breaking. We don't know yet if there's breakage happening that in the fibers or if they're just actually actively retracting. So we can do this two-photon imaging experiment and then process the tongues for whole mount imaging afterwards, so we can kind of get a fuller picture of what's happening both with the gustatory nerves, which you can image with the red channel, and also use the troma staining to image the keratin 8 taste receptor cells. And in an intact mouse, the buds are nice and compact. They have this really beautiful structure. But here's an example of a bud that was after treating with cyclophosphamide. You can see that the taste bud is kind of exploded out like a blooming onion, right? The taste receptor cells are kind of disintegrating and flaring out. And similarly, the fibers are also kind of spewing out to the edges. So this is an example where maybe the overall volume of the fibers may not have changed so much, but the morphology has really drastically changed. What's up?
How's the drug is?
This is IP injection. So we followed this over a little bit longer time point. So up to now, we've been focusing primarily on this early, day 4 period where the most reduction happens. But if you keep imaging for more days, you can see that the fibers recover, and even maybe at day 16, there's an over-innervation phenotype where you have more fibers innervating the buds. And then this eventually kind of goes back to normal after 20 days. So this is just a quantification of this at these different time points, showing that the most interesting thing to me is if you look at this day 20 region, although there's no significant difference between the starting and the ending volume items, it's like the buds have lost their character. Like smaller buds have become bigger, bigger buds have become smaller. And so on average, there's the appropriate level of innervation, but within each bud, it's kind of they've forgotten who they are. And they become kind of a big bud becomes a small bud or a small bud becomes a big bud after that point. So we find that cyclophosphamide induces a transient retraction. We think it's a retraction. It could be a breakage, but likely a retraction of gustatory fibers from the taste buds, and that these individual taste buds vary in the severity of their fiber of loss and also their taste receptor cell loss. But since gustatory fibers produce trophic factors that can promote taste progenitor proliferation and differentiation, perhaps trophic factors could also be used as therapies to improve the taste function, either during or after chemotherapy. So open questions, we don't know whether or not radiation therapy also affects gustatory fibers. We know from previous reports from many labs that the radiation also affects the proliferative capacity of these taste progenitors. And we also don't know what the time course and mechanism of recovery of taste synapses is, and how it might functionally change the taste signaling after chemotherapy. So people have reported, right, they have persistent taste dysfunction, even though in the mouse models, mostly the taste receptors are completely recovered. Fibers, at least on average are recovered, but people still experience prolonged taste deficits. So where is this, taste dysfunction arising from? Is it a failure of fibers to find the appropriate taste targets in the bud, even though they are there? Is it a not full recovery of function of the taste receptor cells? I think these are still open questions that we're really excited to pursue in the future. So I know we're getting very short on time. So I think you guys probably looked at I heard that some people did a journal club on this paper potentially. But one of the questions that we had was to determine whether gustatory fibers are kind of driving formation of synapses in the taste bud, or whether they are following cues from the taste receptor cell to form a synapse. So I've been teaching developmental neuroscience for my graduate students, and we study the neuromuscular junction, right? And there's always this kind of play between the target tissue, the motor neuron, and the muscle cell, and they give each other signals, right? So I was trying to think about the neuromuscular junction in my mind when I was thinking about this problem within the taste field. And so I think we have, now some tools that we can use to answer some of those questions, at least a little bit, right? And some of these, as you might know, are the CalHM1 protein, which is expressed at these presynaptic sites within the type 2 cells, and bassoon, which is the presynaptic marker for type 3 cells. So we have kind of two different synaptic markers, CalHM1 and bassoon. And they're tricky to use in the lab, but we've got good protocols working so that we can use immunohistochemistry to identify these presynaptic markers, along with the gustatory neurons. So we typically use a P2X2 antibody to label gustatory neurons. We've also done this with the FOX2 AI14 lines to label those transgenically. And then, of course, we use Troma for just the mature taste receptor cells to label those. So this is just an example of immuno that's looking at CalHM1, and we can look at where these puncta are and how close in proximity they are to gustatory fibers labeled in red. So we've gone through lots and lots of images to quantify these things, to see how many might be just on the surface of the taste receptor cell, but may or may not be contacted by a gustatory neuron. So if we found that lots of these puncta were kind of on the cell, but not near a gustatory fiber, we would kind of conclude that probably the taste receptor cells making those presynaptic sites and then either attracting the gustatory neuron, or at least, it's happening first. But what we found was that the vast majority of these presynaptic puncta, either bassoon or CalHM1, were contacted by gustatory fibers. So over 90% were contacted by a gustatory fiber. Which isn't 100%. Obviously, it's not always that they're always connected, but it's a high enough percent that it looks like gustatory fibers probably are more, they're likely involved in this thing. So to get at the crux of that question is, it's likely necessary, but we've got this population. So what happens, right, if you take away the gustatory nerve innervation? And so what Shannon did was just do some chordotympany nerve cuts or glossopharyngeal nerve cuts to see what would happen to the presynaptic sites once the neurons were gone from the bud. And so in order to do this, we first needed to assess when those gustatory fibers would disappear within the bud, but do it at a time point before, as we know, the taste receptor cells would die off on their own because of lack of trophic factors. So there's kind of like a Cinderella or a Goldilocks point right here, where we would have to have the gustatory fibers be gone, but the taste receptor cells still intact enough to show whether or not they would have this contacts. So we found that at day 4 post-transection, both for the glossopharyngeal and for the chordotympany, that was the point where we could reliably have a loss of all the gustatory innervation, but still fairly intact taste receptor buds. And we have quantification of how many taste receptors were lost at this point, but I'm not going to go into too much detail because of time. So long story short, we see like massive reduction, right? It's not zero, but it is close to zero for bassoon and for CAL-HM1 fungiform buds. The presynaptic puncta almost immediately disappear by that day 4 time point. So it shows that these presynaptic sites are lost. And so we think that the normal accumulation of presynaptic machinery is really dependent on nerve fiber innervation. But what happens when the nerve regenerates? So we have this great tool, right, in the peripheral nervous system that we can regenerate the taste fibers if we give it enough time. And so we used our imaging technique to find out when an appropriate time frame would be to look at this process. We see that the fibers go away primarily at day 4, and then they start to recover a little bit at day 8. So we thought, that's the day 8 to day 14 is kind of the prime time to look at the reinnervation. And you can also notice here that we get this over-innervation phenotype, just like we did with the cyclophosphamide. So I think damage to the nerves at some point then induces this hyper-innervation type of phenotype that we're seeing. And we're seeing it in different contexts, but it seems to be kind of a recurring theme. So we confirmed this with the immunohistochemistry, and now we're looking over time. So at this important day 8 time period, we can start to see bassoon was returning. There's a couple of little puncta here and there. But if we quantify this, at day 8, there is a wide variety of the types of puncta, right? Some are contacted, some are not contacted within the buds. And you get this kind of distribution of these puncta that may or may not be contacted by the nerve. So this is really interesting to me because I think it indicates something about that it's not the physical contact of the gustatory neuron onto the taste receptor cell that is recruiting these synaptic puncta, or at least coalescing them into what we can call a presynaptic puncta. But there's something, right, about the presence of this neuron that's inducing the taste receptor cell to start accumulating these presynaptic sites. And this is true for bassoon and for CAL-HM1 in the fungiform buds. We haven't tried in the glossopharyngeal just because we've heard that it takes a really, really, really long time to reinnervate the glossopharyngeal. So we haven't been patient enough to do it in that system. So I know we're out of time here, but the gustatory neuron innervation, I think, is really necessary for the formation and maintenance of presynaptic sites. And it's dependent on the proximity of gustatory neurons, but not direct contact. So this indicates to me that signals from gustatory axons are helping to organize the presynaptic proteins in the taste receptor cells to make functional synapses. And so now the big question is, what are these factors? And can we use this mechanism to help promote or improve taste function for those people who have taste disorders? So thank you very much for your time. And I apologize for all of the disruptions of the talk, but I've been really appreciative of your questions, and I'm happy to answer questions if you have time.